Okay, so our next lecture is going to be on our extracellular matrix. Our extracellular matrix, of course, is going to be very important. So we're going to talk about the different components and how they work together to provide extracellular matrix functions. So the extracellular matrix is what's going to help bind your cells together, and they're going to have other functions such as regulation of cell activity. They can regulate adhesion of cells, migration, proliferation, differentiation of cells. So it's a very intricate network of macromolecules. So it's an acellular matrix. Now they're going to be made up of two major classes, your glycosaminoglycans, or your GAGs, and also your fibrous proteins. And so you're going to have, within your extracellular matrix, you're going to have essential support structures for the cells, and everything is going to be secreted by resident cells. So cells within a tissue will secrete their own connective tissue components, their own collagen, elastin, whatever they're going to be making. They'll secrete them all, the proteoglycans, etc. Now, your extracellular matrix is going to also form a basement membrane or basal lamina. That is a special part or a special version of extracellular matrix. So the basal lamina also is called basement membrane. And this is going to be dense. So this is going to be more dense than your other connective tissues. So it's going to not allow for as much flexibility as your other connective tissues. And usually it's going to be right under your epithelial and endothelial cells. And they also surround muscle cells, Schwann cells, and their goal is to protect against mechanical stress. Now, they're just like other extracellular matrices. Your basal lamina also will have a structural support. It is also going to be involved in filtration, and of course, a surface for attachment of your cells. So in general, your extracellular matrix are going to be a portion that can help cells bind to areas, help cells migrate, which is going to be critical for your immune system. Those are one of the more migratory types of cells. You also have a special type of basement membrane in the kidney, your glomerular basement membrane. And then this membrane is ribbon-like. And this is going to be between the endothelial cells and the podocytes, foot processes. Okay, so again, the extracellular matrix is going to hold the cells together. It's going to be part of connective tissue fibroblasts. The meshwork is made up of these glycosaminoglycans or GAGs, and these are going to be covalently linked to proteins, and they're going to be covalently linked to things like the proteoglycans. Your fibrous proteins are going to be your collagens, elastins, and fibronectin. So the extracellular matrix is going to be made up of three major classes of biomolecules. And these three major classes are going to be your structural proteins, which are going to be the collagen, fibrillin, and elastin. And then we're going to have specialized proteins such as fibronectin, laminins, and integrins. And then the proteoglycans. Proteoglycans are going to have a protein core with long chains of repeating sugars. And the sugars are in the disaccharides. And so the units are, this is going to be what gives them the call them glycosaminoglycans. That's the glycosaminoglycans. These are going to be very complicated. They're going to have high molecular weight components, and depending on what constitutes the extracellular matrix, these are going to allow for spreading, for example, of your extracellular matrix. Now, the important thing about the extracellular matrix is often it's thought about as a glue that holds things together, but it is actually very important in filtrating and also keeping cells in their proper locations and proper organizations. And so diseases associated with defects in this are diseases such as rheumatoid arthritis, osteoarthritis, and many of these are involved with aging as you get damage to the functional capacity of the extracellular matrix to perform its functions, not only of gluing things together, but of filtering things. And they have a role in, for example, keeping bacteria away from your cells, things like that. So the extracellular matrix is a very important part of your tissues. Your connective tissues are basically extracellular matrix and your fibroblasts and other substances, ground substance. Oops. And your ground substance is going to be the components of your extracellular matrix, glycosaminoglycans, proteoglycans, glycoproteins, laminin, and fibronectin, however, not collagen. So you do have specialized tissues. You're going to have collagen, in which your chondroblasts are going to secrete these matrix components. And then you also have bone. And when you have your osteoblasts, are going to secrete the matrix components. Now, collagens are going to be the most abundant proteins in all of the animal kingdom within the extracellular matrix. And so it's a major protein of this extracellular matrix. There are 44 different collagen genes known in the human genome, and they are overall going to generate over 28 different types of collagen. Type 1 collagen is going to be 90% of the collagen made. Type 1 collagen is going to contain 33% glycine and 21% proline and hydroxyproline. Type 4 is important. Type 4 is a major component of all basement membranes. So basement membranes are more dense, and so type 4 are able to form a lattice network. And then, of course, collagens are going to be synthesized by fibroblasts, but they also can be synthesized by epithelial cells. Now, collagen is going to be synthesized as a pre-protein, and then it's going to undergo extensive post-translational modification, as well as some pre-translational modification. The alpha chains of your collagen are going to associate into a triple helix structure, and these structures are called collagen fibrils. These are going to have two identical alpha chains and one different alpha chain. So you have two identical alpha 1 chains and one alpha 2 chain. So you get two alpha 1 chains and one alpha 2 chains. Now, the procollagen or tropocollagen is going to be synthesized on the rough ER. So you have the pre-protein procollagen or tropocollagen is to be synthesized on the rough ER. And then you have a decent table in your book which goes through how collagen is synthesized. And so collagen is going to be synthesized at different locations. So of course, as all you're going to have to go through the endoplasmic reticulum, and then you're going to package up your collagen. Your collagen goes through the Golgi apparatus into a secretory vesicle, and then it's going to go into the extracellular space. And so you have in the rough ER the synthesis of your pre-pro collagen, and this will then go through a portion of hydroxylation of the proline as well as lysine residues. And you also get glycosylation of your hydroxylysine residues. They then, in the ER, as well as it moves through the Golgi, are going to become the tropocollagen molecule and self-assembly into that. And this goes by the formation of the disulfide bonds in the carboxy terminal, and you get the triple helix formed in this space. This triple helix then is going to be secreted, and then you get cleavage of these propeptides, these triple helixes, removing amino and carboxy terminals, and then you get assembly into collagen molecules, into fibers, and then what makes up the extracellular matrix. So just to recapitulate that, we have our collagen in the ER is going to have initially a precursor called the pre-pro collagen. And then this is going to be in the rough ER formed. Three of these pro-collagen molecules associate through the formation of disulfide bonds at their carboxy terminus. And then these three are going to form the triple helix from the carboxy end towards the amino end, and this is going to form tropocollagen. So you have three of these, and these are going to form disulfide bonds. So it lines carboxy to the amino end. And then this is tropocollagen. And then tropocollagen gets secreted from the cells. And then you get extensions. So the triple helix gets modified. The ends get cleaved off basically, and it matures in the extracellular space within the extracellular matrix. So you get maturation. And then you have your fibrils of collagen, which will line up in a very organized manner and form collagen fibers. And each collagen alpha chain of a fibril has about 1,000 amino acids associated with it. Now, collagen also has a very unique amino acid sequence. Oops. Now you're going to have hundreds of repeats of glycine, proline, and something else. And that X can be something else. Or you're going to have glycine, X, and your hydroxyproline. Now, overall in a collagen molecule, approximately 35%, 33 to 35 is going to be glycine, somewhere in the 30s. And then approximately 20 upwards to 25 is going to be proline. Lysine is also going to be in there, so lysine is often that X. So lysine is also an important component of the collagen molecule. So let's just go over a few of these. So glycine, so your polypeptide chains, those 3 chains, are going to be linked together by hydrogen bonds. And each turn contains 3 amino acid residues. So as collagen winds, 3 amino acids are at each turn. Okay, so in this case, every third amino acid that is in close contact with the other 2 strands in the center, and only glycine can fit into this position. Only glycine can be flexible or not interactive as much to make sure that the collagen winds properly. So there are going to be 3 amino acids at each turn. The third amino acid is in close contact with the other 2 strands. There's alpha 1, 2 alpha 1s, and 1 alpha 2. So those are the 3 strands. And so only glycine can fit this position. So that's why, regardless of lysine, regardless of proline, hydroxyproline, glycine always has to be that third amino acid. So you'll have another glycine here, etc. And that's because the glycines can't interact without the structure collapsing. So again, you have your glycine with 2 other repeats. Typically, your repeat is going to be proline or hydroxyproline, but glycine is always going to be that third position. Lysine will also find its way in there, and there's a purpose for lysine as well, which we'll talk about. So hydroxyproline residues are going to be involved in hydrogen bonds. And you can see here that we have vitamin C. So we have proline residues, oops, and with an alpha-ketoglutarate, we have a hydroxylase with vitamin C will generate a hydroxyproline residue, release succinate. We also have our hydroxylysines, which is the same sort of thing. You have your hydroxylysine, which is going to have a site of attachment for galactose and glucose moieties. And you're going to, again, start with a lysine residue, requires vitamin C, and then you're going to add that hydroxide group through the hydroxylase, and you're going to release succinate again. And then what's important is cross-linking. So side chains of lysine will be oxidized, and they can form aldehydes and something called lysine, which is an intermediate. And why this is important is because your aldehydes can condense with amines. So your lysine residues through oxidation will become a aldehyde, and then this can condense with a second lysine residue, and you can form what's called a Schiff base. Now, a Schiff base is just simply formed by the condensation of a primary amine and an aldehyde and it involves cross-linking of these two groups. And what's important for that for your extracellular matrix and making a shift base is that it can help form hydrogel networks. Okay, so we can also have another your aldehydes can also condense. So you can have two allosynes, and they can condense, um, and they can have a similar result in the condensation is helping to form a hydrogel type of um, um, in this lysine or leucine formation. And so the formation of these crosslinks in collagen are important. They're going to be covalent crosslinks between different collagen molecules that condense into these shift bases, for example. So lysine residues are oxidized to that allosine, and that reacts with the amino group of a lysine residue on another molecule that forms that shift base. Or you can have two allosines on different molecules that can undergo aldol condensation. And so you start to form a network. All right, so then collagen fibers are going to be formed. Mature collagen fibers are formed in the extracellular space. And then this is going to require lysine and hydroxylysine residues to be oxidized by that lysyl oxidase. And then you're going to have collagen peptidases, which are going to remove both the N and C terminal domains of each of the fibrils that are released. Okay, so now there are at least 19 types of collagens have been characterized. Of course, your body makes more collagen, but they're in such small amounts that they haven't been characterized, but 19 have been characterized. They can be classified as being fibril forming. They can be classified as being associated with fibril surfaces. Some are going to be transmembrane proteins, and some of them have beaded filaments as well. But in all, collagen are going to contain 3 polypeptide chains with at least one stretch of a triple helix. The non-triple helical domains, they can be short or large. One of them that's large is a network forming collagen type 4 that creates a mesh-like structure. So collagen are going to be found again in transmembrane proteins on the epithelial and epidermal surfaces. Type 1, 2, and 3 are going to form fibrils that are going to assemble into large insoluble fibers. These fibers are going to get cross-linked through covalence, and usually those are between the lysine residues. As we just talked about, forming shift bases. And then these are going to, cross-links are all going to give them their different types of structures. So some of these structures are going to be, oops, sorry. You have your tendons. I'll just give a few examples. So you have tendons that are going to contain collagen fibrils that are lined parallel to the long axis of the tendon. And this gives the tendon a lot of tensile strength. And then we also have, again, our fibers, such as our type 4, that are part of your basement membranes found in many tissues, of course. And they are going to form a flexible collagen. And so that's going to be due to the collagen type 4 are able to form tetramers. So it's going to be more flexible than what you find in your tendon, for example. And so you get that lattice network that is shown there. Another component of the extracellular matrix is elastin. So this is going to be a major component in elastic fibers. And so you can imagine that these are going to be in the connective tissue of smooth muscle cells. We also have our endothelial as well as our microvascular cells. Our chondrocytes and our fibroblasts as well. Oops. So elastin basically, in essence, does what it does. It has elastic capabilities. It can allow the tissues to expand and contract. As we all kind of know. So this is going to be very important in your blood vessels. It's also going to be very important in lung tissue. Elastic fibers are going to have microfibrils, and these are going to be composed of acidic glycoproteins. especially fibrillin 1 and fibrillin 2. Now, elastin is, of course, going to be made up of a pre-protein called tropoelastin. It's kind of got stretched out. So for tropoelastin, there's only one gene. Elastin is going to have a lot of cross-linking. It's going to be very insoluble, and it's going to have an amorphous structure. The tropoelastin has high solubility. And it's going to contain two types of altering domains. It has a hydrophilic sequence-rich lysines, and alanine. So, K-rich. Let's say alanine domains. Okay, and so basically you're going to have lysine cross-linking domains that are going to contain proline, which are called Kp. So you can see here's our Kp. Cross-linking. And then we have a lysine cross-linking domains with alanine equal Ka. So we have our Kp cross-linking and we have our Ka cross-linking. And then interspersed between all of these are these white domains or white regions, and these are hydrophobic. So we have our hydrophobic domains here. You also have a hydrophobic region which is rich in valine, proline, or glycine. So when the tropoelastin is secreted from the cell, it is aligned with the microfibrils, and the lysyl oxidase is going to initiate reactions that cross-link elastin molecules, and the cross-linking is again going to use lysine. So lysine is going to be cross-linking. The net result of this cross-linking is the generation of a fibrous mesh that encircles cells. Now, elastic fibers do have the ability to stretch and then reform, and they do this without requiring outside energy source. And the stretching So when elastic fibers are stretched, the amorphous elastin structure in all gets stretched. And this is really going to expose those repeating hydrophobic regions, which is critical. And of course, this is in an aqueous environment. So naturally, the stretching exposing these repeated hydrophobic regions is going to lead to a decrease in the entropy of water. And then when the stretching force is removed, elastin will naturally go back to its original structure because the hydrophobic effect is the primary force that allows the structure to reform because those hydrophobic regions want to hide again from water. Okay, and so elastin is very stable, has a half-life of about 70 years. So it has a very, very long half-life. Now, another one is laminin. So we talked about the basal lamina, or the basement membrane, and that type 4 collagen is the major component. After type 4 collagen, laminin is the next major protein found in the basement membrane or basal lamina. Oops. Okay, and so laminin is actually going to provide structural support, and it binds to the type 4 collagen. As well as other molecules, and it's going to also bind to cell surface associated molecules, the integrins. So not only do the extracellular matrix bind to each other, but they are also going to interact with the cells in the tissues and bind to them as well. So this is a laminin structure. It's a heterotrimeric protein, and it's shaped like a cross. It has laminin 1. It has alpha, beta, gamma subunits. So this is laminin 1. We have our alpha, we have our beta, and our gamma chains. And the major feature also of this is going to be a coiled alpha helix, which joins the 3 subunits to form sort of a rigid rod. The laminin extensions is what really allow it to interact with other components. The extracellular matrix components, such as collagen, lipids, proteoglycans, will all bind to those alpha, beta, gamma chains of your laminin structure. So this is that coiled alpha helix that helps to form what's referred to as rigid rods. And now moving on, we have our bottle brushes, our proteoglycans. These are very important in forming gel-like structures in the extracellular matrix. They consist of the polysaccharides, glycosaminoglycans, linked to a protein core. Now these are going to have repeating units of disaccharides, and one of the sugars of the disaccharides is going to be an acetylglucosamine or an acetylgalactosamine. Hopefully you remember those from earlier in biochemistry. And then you're going to have either the second is going to be usually an acidic, like a gluconoride or an iduronic acid. So it's repeating disaccharide units. And so you have, we'll say X and Y, and X is going to usually be an acetylglucosamine or an acetylgalactosamine. And then Y is usually going to be acidic. And these will repeat XY, 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 etc. So the sugars are going to be modified by the addition of sulfate groups to the parent sugar. And the sugars, the proteoglycan can consist of up to 95% oligosaccharide by weight. The charges, so the carboxylate and sulfate groups, are going to have negative charges. Will bind to the positive charges. And this is going to form hydrogen bonds with trapped water, and this creates a hydrated gel. Okay, so your proteoglycans are going to provide flexible mechanical support and also act as a filter. So your proteoglycans are going to be found in your interstitial connective tissues, such as the synovial fluids of joints, the vitreous humor of the eye, atrial walls, cartilage, bone, your cornea. They're going to interact with collagen and elastin, fibronectin, whatever's there, and also to laminin and laminins there. So they're going to have many different repeating chains of glycosaminoglycans. And this the glycosaminoglycans, they have another name, which is the mucopolysaccharides as well. So again, they're going to carry a negative charge, they get hydrated, and so they're going to act as lubricants. Now, here's some examples of our repeating disaccharides. Some of these are going to be well known to you. Hyaluronate is very important. Heparin is important. Keratin sulfate, dermatan sulfate. And so you're going to have an acetylated sugar with that acid, and then the sulfate groups, again, are also going to be present to add more negative charges. So even though these are kind of blurry, you can see that they have negative charges associated with all of these. Some of them will have sulfates in addition to that. And we have our dermatan sulfate, which has our sulfate groups, etc. So basically, these guys are just formed so that they can interact with the environment. Another thing that is going on with your proteoglycans is that you have long negative charges because they're repeating units, and these are going to, of course, repel each other. So this is another important aspect, because this means that your proteoglycans are going to occupy a very large space. And this is going to be important for giving flexibility to cartilage, permitting expansion, re-expansion in those areas as well. There are at least 7 types of glycosaminoglycans that we just talked about. We have our heparin sulfate, hyaluronic acid, etc. And all of them except for hyaluronic acid, the glycosaminoglycans are going to be linked to either serine or threonine residue. Oops. Now for hyaluronic acid, this is what is going to give the ability of tissues to resist compression by providing Tuger force or swelling force. And this is its ability to absorb significant amount of waters. And so your glycosaminoglycan is going to, in this case, be bound to your sugars, xylose and galactose, as shown here. And so this is going to be important in load-bearing joints. Okay, so your book has this table and kind of goes over the function sum of the the proteoglycans, glycosaminoglycans. So you have hyaluronic acid, which is going to be important in migration. So it makes sense that it's going to be involved in your immune system, in wound healing, etc. We have our heparin, which is an anticoagulant, can bind to thrombin 3. And then we have heparin sulfate, for example, which is going to be important in skin fibroblasts, etc. Now your proteoglycans are going to get synthesized. So the protein component, of course, is going to be synthesized on the endoplasmic reticulum. So you have here your protein core, which are these little purpley things here. And so this comes from the ER, of course, because that's how you make your proteins. It's going to enter the lumen, and this is where glycosylation is going to occur. And again, we have those UDP sugars, which are going to be our precursors. The UDP precursors are going to be added one at a time. The first one is going to be added to the protein, and then you're going to add the sugars to non-reducing ends. The glycosyl transferases are going to be specific for the sugar being added. The type of linkage that forms are going to be with the sugars already present, of course. Sulfation occurs after the addition of sugars. And so you can get your sulfate groups, which are shown here, added after. Now you have 3' phosphoadenosine 5' phosphosulfate, or PAPS. This is also what's called active sulfate, and this is what provides the sulfate group. Let me write what it is first. Okay, and so PAPS is what provides the sulfate group. So it's also called active sulfate. And so the other thing that you can have is gluconic acid can be converted to ioduronic acid, and this is through an epimerase reaction. Okay, so in essence, you're basically going to build your proteoglycan starting with that protein core, and then you're going to add your UDP sugar. You're going to have a xylose residue. It's going to be first. So you get your xylotransferase, that's for that first position. And the second one coming in is going to be a galactose, shown here. And so you have your galactose transferase. And then your third position, you're going to have another galactose added. That's shown here, and that's through a galactotransferase 2, etc. So you just start building all your sugars. All right, and so basically the structure of your proteoglycans are going to resemble a bottle brush. So that is what's shown here. So you get a bottle brush looking. And so again, you have your protein core, which is shown here, and then you have your repeating disaccharide units here, and they're just going to repeat. They can form large aggregates, and they can have an attachment or link um, to hyaluronic acid, for example, shown here. You can also have other proteins, keratin sulfate, chondroitin sulfate, etc. So hyaluronic acid will be sort of the core protein, and then, or hyaluronic acid will link the core proteins to this, and then you'll have your bottle brush or your repeating disaccharide units that form these sort of bristles, if you want to call them that. Now, proteoglycans are going to interact, of course, with other components of your extracellular matrix. And so you're going to have cross-linked fibers of collagen associate with this complex. You're going to have your bottle brush proteoglycans, which sort of spread things out, interacting with fibronectin. On the cell surface, you're going to have integrins. Integrins are what are going to interact with your extracellular matrix. And the extracellular matrix is going to help to keep cells in place or help cells move through the matrix, etc. So they're all going to interact together. Now, one thing that's important also is you have to degrade proteoglycans. And so degradation of proteoglycans is going to occur within lysosomes. So lysosomal enzymes are going to degrade proteoglycans, glycoproteins, and glycolipids. So your proteoglycans are going to have to go to lysosomal compartments. And so any deficiency of lysosomal glycosidases are going to lead to partially degraded carbohydrates. And then the glycoproteins and glycolipids can accumulate. And this can lead to what are referred to as residue bodies. Now, another thing that can happen, well, first of all, the accumulation of these partially degraded glycosaminoglycans are going to lead to mucopolysaccharidosis, and this can lead to deformities of the skeleton. And it can also lead to mental retardation as well. Now, another thing that can happen is you can have over degradation of your proteoglycans. Remember that your proteoglycans are going to be made in your cells as a preform, and then it gets secreted into the extracellular matrix. You can have products get degraded inside of the cell, or you can have your products get degraded outside of the cell. And so inside of the cell, it's thought that you have a half-life of about 4 to 30 days. Outside of the cell, that half-life increases enormously. However, in some diseases such as systemic lupus or erythematosus, SLE, degradation is going to be favored. So these half-lives will be much shorter in a person suffering from SLE, for example. Okay, so moving on to our integrins. And our integrins we've got introduced to just a little bit. These are going to be glycoproteins that attach to the cells, to the extracellular matrix proteins. So these are cell-bound, and they attach to the extracellular matrix. Now these are integrin families are going to be very important not only to development, so for embryogenesis, to our immune responses, but also these are going to be important in cancer metastases and tissue homeostasis. So when a cancer develops, the cancer stays tethered to the extracellular matrix and it stays within the organ in which it's developing. When you get a breakdown of that tethering, then the cancer can metastasize and can be moved throughout the body. So integrins are going to contain a large alpha and small beta subunits. So here's our integrin here. So they're going to have they're going to be subunits. The alpha subunit is what's going to determine the specificity. So it tells basically alpha is what will bind to the laminin, for example, or whatever it's going to bind to, but it's specific for that. So for integrins, the extracellular components that can be bound are going to be things like fibronectin. Space, okay. Collagen, laminin, vitronectin, glycoproteins. One important one that it does bind is something called tenascin. Etc. So these integrins can bind to many things. In this picture here, we have this particular integrin binding to fibronectin in the extracellular matrix space, but it could be collagen or something else as well. So another function of integrins is going to be anchoring cells to the skeleton of the extracellular matrix, as well as those that are associated with your immune response, and they allow your immune cells to attach to a site of infection, for example. So if an infection occurs, your integrins get activated on your white blood cells, and here we show on our leukocyte we have something called L-selectin. And you also are going to have carbohydrates on your immune cells. And so on your inflamed endothelial cells, they're activated. They're going to also have their integrins upregulated. Here we have P-selectin and E-selectin. And so when an infection occurs, your immune system as well as the site of the location are going to get activated, and it's going to cause your immune cells to know where the infection is so they can bind to that area, enter it, and hopefully control the infection. So the activation of integrins are going to enable the migration of your white blood cells. So during an infection, your endothelial cells get activated. We'll talk about this more in immunology. And then they're going to increase integrin expression. And so that's that P-selectin, E-selectin. And this is sort of tells your immune system where the infection is. So for example, if you have a coronavirus infection of your lung cells will increase expression of European E-selectins, and it'll tell your white blood cells that are circulating that this is where the infection is occurring. You need to stop, enter the lungs, and try to get this infection under control. And so that's another function of integrins. So all of this is leading to adhesion proteins, and adhesion proteins, what we've kind of been talking about. The extracellular membrane is going to link to cell integrins, and this is going to allow the cells to interact with the extracellular matrix as well as stay in place and do whatever they need to do. Adhesion proteins, fibronectin is an example, are large proteins that are going to be binding to many different components simultaneously. And so it's just what you have thought it was. You have your integrins, you have your collagen fibers, you have laminin, and your integrins are going to be interacting with your collagen, laminin, etc. And these are all going to be important in just overall adhesion. So this is providing a bridge between the cytoskeleton of the cell via actin, as well as the position of the cell within the extracellular matrix. So all of these are going to be linked downstream to dystrophin. We have talin here, vinculin, and these are all tethering to the actin filaments. So the signals from the extracellular matrix are going to change the shape of your cells depending on what they are. So again, going back to the immunology example, because that is one of the more active or changes in cell structures. So when you do have an activated endothelial cell that thinks that there's some infection going on, the structure of that cell will change so it becomes leaky. It allows movement or migration of your white blood cells and leakiness of your red blood cells, of course, because they're just there, into the actual tissue. So it's going to change the shape. And the reason it can do that is because it's going to be, the signals are going to be connected to the actin and the cytoskeletal structure of the actual cell. So just a little bit, we have our metalloproteases in the extracellular matrix. And we've already talked about these, but just as a reminder, they're going to be containing zinc. They are important for cleaving all the proteins found in the extracellular matrix, and that includes those collagen fibers, the laminin fibers, so they're all going to be zinc proteases. They're also going to be important for degrading the extracellular matrix proteins, such as your proteoglycans and things of that sort. Now the mucopolysaccharidosis, again, these are where you have defective enzymes in breaking these down. You have a series of them. And you can see here that the products that are accumulating, are those products that we just talked about. Proteoglycans, you have your heparin sulfate, you have your dermatan sulfate, a different color. And so these are going to accumulate, and there are various deficiencies of the different enzymes, and the diseases are given different names. Sanfilippo A is going to be the most severe. And so you're going to have missing or alternative forms of heparin and sulfase. That's that heparin sulfaminidase. And then Filippo B is when you're missing or you do not produce enough of your alpha and acetylglucosaminase. And then D is when you don't produce of the 6-sulfatase. But the Sanfilippo A is definitely the most severe of them. And that is the end.